So today I'm going to cover the remaining part of the seventh unit. In the first part, we have learnt about the active packagings and the definition of intelligent packagings, and I explained about active packaging in detail. Okay, so today I'll cover the intelligent packaging. So before starting the intelligent packagings, I'll brief you about what is the smart packaging is the sum of these two techniques, that is the intelligent packaging and active packagings. So smart packaging provides a total packaging solutions that on one hand it monitors the changes in a product or its environment, and on the other hand it acts upon these changes. For example, the scavenging function involves the collection and removal of unwanted gases or odors from the pack, but smart packaging technique can also be used to release the additional aroma into food to enhance the freshness and the flavors. Fine. So from these slides you can take that smart packaging is a set of both active and intelligent packagings, in which we have grouped the function or the, you can say, the features of both packagings, like intelligent as well as active packaging. So now I'm coming to the intelligent packaging, and in the bottom you can see there is some explanation about PETG that I forgot to define in the last lectures. So from slides you can read it. Coming to the intelligent packaging, though we have already covered about the definitions, so I am again here repeating. Intelligent packaging is what? It is defined as packaging that contains an external or internal indicator to provide information about the aspect of the history of the package or the quality of the food. Simply mean, we will say this is a intelligent packaging if there is a indicator either inside or outside, and is this indicators provide the history of the package. For example, when it was packaged, or you can say whether it is appropriate to use or not. If we talk about the quality of the food, it means whether it was appropriate to use or not. And history of the package mean to say that for how long, at what temperature, and how long, for how long time it was kept on the in any storage conditions. Fine. So in this picture you can see there are the two boxes, in which here it is written stale and here it is written fresh. So in this we have kept some food products. These are some cut vegetables, and on which we have put a adhesive label, and it is appearing like yellow. With the passage of times, there will be some biochemical changes in the food product that will be sensed by, or that will be detected by the this sensor, or sorry, this indicators, and they will bring out some changes. And due to this, you can say, volatile compounds, those are emitted by these food products, this indicator will turn its color from yellow to bluish, and this will be called as stale kind of, that is not fresh, means to store for last 4-5 days. So this is the concept of intelligent packaging. Intelligent packaging system can be classified into three categories. What are these three categories? Number one, indicate product quality. Number one, indicate product quality. For example, quality indicators. It can be a time temperature indicators, and it can be a gas concentration indicators. Number two, provide more convenience. For example, during preparation and cooking of the food. Number three, provide protection against the theft and counterfeiting the and tampering. So these are the three, you can say, systems in case of intelligent packaging. So now I'm coming to the first one, indicate product quality. How we can use intelligent packaging to indicate the product quality. So if we talk about the indicating the product quality, the first thing is it can be a quality or freshness indicators. So we can use some kind of indicators. They will indicate the quality or the freshness of the fruits or vegetables. So they basically work on two mechanisms. Either they detect the volatile metabolites such as diacetyl amines, ammonia, and H2S produced during the aging of the food. So either they will sense these compounds and will show a change in color, and that change in color will be irreversible, and based on the change of the colors consumer will decide whether this fruit or vegetable is acceptable or not. Other mechanism it can be they can sense or they can detect organic acid or volatile biogenic amines formed by microbial degradation of the protein containing food, such as patricins from the arginine, cadaverine from lysine, and histamine from the histidines. So these are the biogenic amines. What are the biogenic amines? Histamine is the biogenic amine, cadaverine is the biogenic amines, and patricins are the biogenic amines, and they are formed by the microbial degradation of the protein, and these are the amino acids, and from these amino acids these biogenic amines have been formed, and it can be detected by the indicator, and then they can change the color. Very popular one, this is the ripe sense indicators, ripe sense sensors developed by the New Zealand. Okay, and it has three variations, or you can say three color zones. Like if it is indicating red spot, then red spot is indicating that the fruit inside the package it is crisp. With the passage of time, it will the crispiness will be slowly loosens, and it will be become more firm and little juicy, fine, and then it will become kind of uh, pale yellow kind of, or you can say brownish yellow, and finally this juice content will increase, and then it will appear a very yellow spot. So if it is a yellow spot, then we can say that it has attained the full maturity and it is very juicy. If it is red spot, we can say it is a very crispy. So this sensor, how it works, it detects or it absorbs or it senses the alcohol, esters, aldehyde, ketones and lactone that contribute to the aroma. For sure, as the maturity or the ripeness of the fruit will prolong or will exceed, then this there will be a emission of aromatic compound, and on the basis of aromatic compound detections, this color change will take place. So this is the quality or the freshness indicator. Now I am coming to the next type of indicators which indicate product quality. These are the time temperature indicators, and basically they are used for the frozen foods. Because in case of frozen food, generally we keep the temperature minus 18 degree, and a slight variation in the temperature can affect the quality of the frozen foods. So that's why they are primarily used for the frozen foods. This time temperature indicator. Time temperature indicator simply means they will tell us for how long we have kept the product at a particular temperature. Let the requirement was to keep the product at minus 5 degrees centigrade, but due to some electric failure or due to mishandling, the product attained a temperature of 10 degree, 15 degree or more than that. So these are the indicators. They will tell us if there is any temperature abuse during the handling, or they will tell us what was the overall temperature variation during the storage. So that is known as time temperature indicators. So as I said initially, they were used for the frozen food, but nowadays they are also used for the other products. Now I'm coming to the definitions. These time temperature indicators are the devices which integrate the exposure to temperature over time by accumulating the effect of such exposures and exhibiting a change of the color. What they do simply, they integrate the exposure to temperatures. Means like we have started the storage on Monday and on Friday we wish to check. So it will integrate what was the temperature from Monday to Friday over the time. So time will be Monday to Friday, and by accumulating the effect of such exposure and exhibiting a change in the color. Let our temperature requirement was minus 5 degree, and if it remains minus 5 degree from Monday to Friday, there will be no color change. Fine. But if accidentally if the temperature has increased, then this time temperature will indicate the you can say indicate the disturbance or that mishandling in the storage by a color change. Hope you understand. Further, these time temperature indicators can be divided into two categories. One is the partial history indicators, and do not respond unless some predetermined threshold temperature is exceeded, and another is the full history indicators. So very simple definition of these two. What is the meaning of partial history indicators? Let you have taken two type of the time temperature indicator. One is the full history indicator and another is the partial history indicator. So how this partial history indicator will work? Let the limit of the indicator is minus 5 degree. So if the for a given time or if there is no change, if the temperature remain minus 5 degree, then it will not show any color change. It will show the color change only after the temperature has attained a threshold level, as it has been written here, which do not respond unless some predetermined threshold temperature is exceeded. So it will change, it will tell only if there is any temperature abuse during the storage. Fine. But full history indicators will tell the whole history. Mean what was the temperature on Monday, what was the temperature on Tuesday, Thursday, and finally Friday. So which respond continuously to the all temperature within the limit of functioning temperature range of TTI. So partial history indicators are intended to identify the abusive temperature conditions, and thus there is no direct correlation between the food quality change and the response of this class of the indicators. Fine, because they will tell, yeah, 2 times the temperature has exceeded. That is the job of the partial history indicator. Whether the product quality is good or not, they will not tell. Okay. So these time temperature indicators are based on irreversible physical, chemical, enzymic, microbiological changes, and their responses is usually expressed in visually quantifiable identification in form of mechanical deformation, color development or color movement. Mean to say that what is the working mechanisms or how this time temperature indicator works. They are based on there can be irreversible physical, chemical, enzymic or microbiological change, and their responses means if there is a change, if it's a chemical change or enzymatic change or a physical change in the food product, and that change is bringing the same change in the time temperature indicator, then this time temperature will indicate the response in form of mechanical deformation, color development or color movement. So there are many type of the time temperature indicators, and they are classified on the basis of their natures, on the nature of working. So first type I am going to cover is the chemical type of time temperature indicators. So most of the time temperature indicators are based on the chemical reactions which causes a color change. The rate of the color change increases with temperature because the chemical reactions rate increases with temperature. So as I said, these chemical time temperature indicator are based on the chemical reactions, and these chemical reactions accelerate as the temperature increases. So on the basis of these mechanisms, there are two commercial time temperature indicators which are available in the market. One is the ON-VUTM TTI and another one is the Fresh Check TTI. So in case of this the ON-VUTTI, it is a self-adhesive label. Here you can see these are the two uh, tick of green lines. They are saying that if this blue color appear on the product, then it means it is okay. So it has been pasted on the outside of the product or package. So what actually this label is, it is a photochromic ink based on benzyl pyridines. So there is a ink that is based on benzyl pyridine and which is further activated by UV light. When this ink is activated by UV light, it turns to be blue, which cause them to turn a dark blue color. Okay. So with the passage of the times and the exposure to the temperatures, the color of this dye faded slowly. So this portion is indicating that it is started spoiling, turns progressively lighter as the time passes, and also if and when the ambient temperature rises. So this color changes takes place due to two reasons: with the passage of time as well as if the temperature in between rises. Fine. So ultimately it will fade up, and this it will match the outside shade, or it will be, you can say, even lighter than the outside shade. If this condition happens, then it indicates that the shelf life of the product has gone or it is the end of the shelf life of the product. Second type of the indicator is the press check indicator, and in case of press check indicator, you can see there is a small round ring inside, and this ring is a polymer, that is diacetylene monomers. Fine. So with the passage of time and surrounding there is a reference ring, okay, and this is the printed reference ring, and with the passage of times it turns to be dark, and if it is dark then it will appear do not use, or it is recommended to the customer that if the circle has become dark, please do not use. As the press indicator exposed to the heat, it gradually changes the color to alert the consumer on the op, of optimal freshness. So it will appear do not use if it is dark color. So next I am coming to the physical time temperature indicator, and in this case physical time temperature indicators, in case of chemical time temperature indicator, the mechanism of working was chemical reactions, and here it is physical mechanisms. So the popular one is this monitor mark TTI, and it is developed by 3M company. Probably I think it is from US one. So it has two portions. One is the this black strip, and another is this white portions. So they are generally categorized as part A and part B. So this part A is the kind of pad it has, and in this part B there is a wick or a cloth, porous cloth you can say. Okay, so this has been attached to the food package, and as the temperature increases in between these two there is a polymer membrane which get melted due to the high temperature, and this dye or you can say it contain a kind of dye. This dye start diffusing, so it will indicate it will travel to point 1 2 3 4 5. So to the distance travel it will give a direct relationship that for how long, for or you can say to how much temperature it has been exposed. This is the working principle of this diffusion, is the working principle of this physical TTI. In case of physicochemical TTI, as I said, in this case the mechanism is diffusion. This dye is diffusing, and the diffusion of dye will takes place. I think here it is written everything, you can read. The dye remaining within the pad until the carrier substance undergoes a phase change due to the temperature exposure above the temperature. Indicator response is measured reading distance dye from front has migrated past window indicator. Whatever. Listen, here the mechanism was diffusion. This dye is diffusing. So in case of physicochemical, both mechanism will take place, chemical as well as physical. There will be a diffusion of one component, and there will be a chemical mechanism also. So let's read that time temperature devices exploit the chromatic properties of pH indicators by coupling the pH indicator transition to a diffusive mechanisms. So this is the popular sensor, TT sensors. It is invented by this Avery Denisons, and it is based on a organic acid between the two polymer layer. The base layer is rectangular, a rectangular label, and that include a circular area active zone containing the fluorescent pH indicators. Fine. On the non-active part, the label, there is a reference pink color. When the activation layer is adhered to the lower active zone, the acid start to diffuse, resulting the pH indicator turning progressively from yellow to reference pink colors. What happened in this case? I'll explain with this again. You can focus on the point 5, like, okay, so in the physicochemical type of TTI indicators there will be a pink color surrounding this 5 ring. Fine. And here it will be a different color. Okay, and this is a pH indicators, and it is linked with a color change. Allah, yeah. Chromatic properties of pH indicators. As I said, in case of physicochemical indicators, there will be a pink ring outside this ring or outside this hole. So as the acid, there will be a temperature rise, this acid diffusion will start. In place of dye, there will be a diffusion of acid. So this diffusion is due to the physical mechanisms, and then when it reaches to this pH indicator, it will change the pH of this pH indicator. As the pH indicator will as the pH has lower down, this indicator will change the color and it will turn from yellow to pink. This is the concept of this physicochemical TTI. In case of biochemical and biological TTI, the biochemicals will act same as, you can say, this physicochemical one, and the acid that we have taken in the previous case, now in this case it will be produced by a microorganisms. Like this, such a TTI may comprise a culture medium. So in spite of taking a acid, now we will take a bacterial culture. This is a strain of cold resistance non-pathogenic producing bacteria, lactobacillus. And from literature it is quite clear that it remains inactive when it is kept under up to a temperature of -5 degree. But as the temperature reaches to 10 degree or 15 degree, it starts producing acid. Okay, and when it start producing acid, that acid diffuses and it reaches to the pH indicator, and that pH indicator give a change in the color, and we can you can say judge the that this product is no more safe for consumptions, because the this what to say now, this will this lactobacillus is basically not a spoilage causing microorganisms, but its physiology will be similar to the spoilage causing microorganisms. It will be assumed that if it is growing, then the other spoilage microorganism will also grow, and by the color change we can easily detect the whether the product is safe for consumption or not. One advantage of such monitoring system is that the process which cause the food to deteriorate are same kind of those occurring in the TTI, as I said earlier, thus allowing the indicator device to accurately mirror the detect deterioration of the food. So the checkpoint TTI is the example. It is which said visual indicator tag system AB, was previously known as I-point time temperature monitor and is a full history indicator. It is based on the hydrolysis of lipid substrate glycerol trihexanoate in fluid suspension by primarily lipase enzymes. So basically this is the enzymes which act on the lipid and it produces an acid, and that acid diffuses. As a result, it brings a visual change. So these were the time temperature indicator, and third is the gas concentration indicators. So in case of gas concentration indicators, these are basically tell us what about the gas concentration in the head space or inside the product. So they work basically on two mechanisms. Either they will be colorimetric or there will be a luminescence. Fine. So if we talk about the colorimetric indicators, then the popular one are the ageless eye and another is the wonder sensors. Both are from the Japan, and the high cost and lack of reliability due to their reversible nature is limitation of these colorimetric indicators. So the main difference between this ageless eye and this wonder sensor indicator is that it is ageless is the tablet type of indicator, and this wonder sensor is a paper type of the indicators. Okay. So in case of what is the how it works basically, if oxygen is in the limited amount, okay, then it will appear pink. If it is oxygen is less than 0.1%, if the oxygen reaches more than 0.5%, then it will turn blue, pink to blue. In case of ageless eye indicator, the presence of O2, yeah, here it is a problem with this ageless eye indicator. I'll explain later, but in case of before that I'll explain this wonder sensor. Here again it will remain pink if the oxygen concentration is less than 0.1%, and it will turn to blue if it is increases to 0.5%. Okay, but the problem here is that the presence of O2 is indicated in 5 minutes or less. See this, let it is pink at a concentration when it is when the oxygen concentration is less than 0.1%. As it reaches to 0.5%, it is turning to blue. So it has been observed that this oxygen concentrations, that is 0.1%, and if it increases to 0.5%, it can be indicated in 0.5% in the 5 minutes. Mean to say that if we do another kind of physicochemical examinations, so it will take only 5 minutes to speak that the oxygen concentration has increased. But the problem with this indicator that it take almost 3 hours at 25 degrees centigrade to turn from pink to blue. Hope you understand. Mean to say that the reactivity is very less or very poor, and it will be even very poorer than this, because if we store it at a chilled temperatures, at a chilled temperature, the response times are much longer. Hope you understand it. So how this colorimetric sensor works basically? This colorimetric sensor basically works on a redox dye based indicators. So you might have performed the experiment in the microbiology lab, that was the methylene blue reductase test, MBRT test. In that test we add a dye, fine, and the dye color was blue, and we keep the milk in the water bath, and after some time there was a color change, or there was a dye color gets fade with the passage of time in some of the milk sample, and in some of the milk sample it persisted. Okay, then I explained that the reason why this, in some sample there is a depletion in the color of dye, and in some cases the dye color persists. Then I explained that it was due to the redox potential, and redox potential is something that is contributed by the oxygen concentration of the product. So if microorganisms are available in the food product, then the the redox potential will be high. Fine. So these dyes are stable at a particular redox potential. As the redox potential decreases or it alters, they lose their characteristics. So in case of MBRT, reason was that if the milk was raw milk and it is means it has some microorganism, those microorganisms will consume oxygen with passage of time. As a result, oxygen will decrease and the dye color will disappear. So same is the story here that I am explaining here. So these are working on a redox dye based indicator. So redox dye methylene blue, same dye that we are using in the milk as a strong reducing agent, as a glucose in इसमें actually there are two things. One is the redox dye methylene blue, and another is a strong reducing agent, that is glucose in the alkaline mediums. So in the presence of significant level of oxygens absence, if it is a absence of significant level of oxygens, most of the dyes lies in the reduced form, colorless state, and it will be a leucomethylene blue. So if the oxygen is less, this dye is colorless, fine. But when it get oxidized, when the medium is oxidized, it gives more colored form of the oxygen. Typically, if the oxygen is more than 5%, most of the dye lies in its oxidized highly colored form. Simply means that if the oxygen is less, this dye will remain colorless. If the oxygen concentration will increase, it will show the color. The reductant is the glucose, and its oxidized state is gluconic acids. Fine. So a non-redox sensitive dye, acid red 52, is usually added to provide a background pink color. Okay, it has nothing to do with the, you can say, redox reactions. So this is a kind of neutral, or you can say non-redox dye, which is giving a pink color. Okay, so this pink color is a kind of inert color, or you can say base color, but as this, it has a dye, and as this dye get a oxygen rich environment, it is turning to the, or it is giving a blue color. Fine. This is the how this gas concentration indicator works. Another story is the luminance based. These were the colorimetric based indicators. The another is the luminance based indicators. So in case of luminance based indicators, we generally take a dye. Luminance based indicators comprise a lumophore. There is a lumophore dissolved in a solvent together with a solvent soluble resin such as fluorosilicon polymers. So ruthenium based metal organic fluorescent dye, this is a dye particularly, it is encapsulated in a 5 mm diameter and 2 mm thick gas permeable hydrophobic silicone rubber. Fine, and that must be attached to inside of the package. So simply there is a ball. That ball is having a thickness of 0.2 mm and its diameter is 5 mm. Okay, and inside the die there is a, inside the ball there is a die. The name of the die is ruthenium based metallic organic fluorescent dye, and what we do, we will expose it to a LED light, and dot is illuminated with a pulsed blue light from the LED that is absorbed by the dot, and red light is emitted and detected by a photo detector, and the characteristics of the fluorescence lifetimes are measured. So basically there is a ball, that ball is having a die on which we will expose, or we will, you can say, we will expose that ball with a LED light, and then it will start emitting red color. So the intensity of the red color will varies as it will uptake, or there will be it will, the, you can say, the reflection or the emittance of the colors will dependent on the oxygens. So it will be more intense if the more oxygen is present. As a layman or as a student of BSc, you can take it like that. Next is the radio frequency identifications. So the radio frequency identifications, RFID, is the use of radio frequencies to read information at a distance with few problems from obstruction or disorientation on a small device known as tag or transponders that can be attached to the object, so that the object can be identified and tracked. RFID devices contain a transistor circuit employing a microchip, an antenna, and a substrate or encapsulation material. RFID tags that simply identify the package are referred to as smart labels. So you might have seen these chips are available in case of electronic items, or sometimes these RFID tags are also available on the clothes. And whenever you do some kind of billing, then with these readers they sense what is the product code, what is the cost. So same type of the chips can be used on the food products. Okay, and it will, this antenna will help us to assess the data. This RFI reader will read when this product was manufactured, at what temperature it was stored, and then we will maintain the data in the computer, and on the basis of this RFID informations we can segregate, or we can categorize, like this product was manufactured 2 months back and its shelf life is 3 months, so let's sell this item first. Biosensors, why biosensors are required? Next tool is the biosensors, and the reasons why we use biosensors is because for example, if a product is contaminated with the microorganisms, and if we wish to determine what is the total number of microorganisms, then practically either we have to go for total plate count, yeast mold count, or means the traditional methods are very tedious and as well as very time consuming. For, I think if you wish to clarify, or if you wish to state that your product is contaminated with coliform or salmonella, then it may need around 2 to 3 days, I guess 3 days at least, to do the confirmatory test. So it is very time consuming. So we can replace this time consuming method by the biosensors. I think if you are aware, then in the last webinars, one of the professor or scientist from Baba Atomic Research Center, he was saying that they have developed some algorithm, and on the basis of artificial intelligence, now on the, they are taking the FTIR images and another images, and then they are what do you say, this calibrating, or you can say interfacing it with the microbial numbers. So now they are simply scanning the product, and on the basis of their physical properties, they will tell that this product is having this much microbial load, and that microbial load will be approximately accurate. Approximately, not accurate, approximately similar to that if we do the physical examinations. So biosensor devices consist of bioreceptors, that is specific for a analyte. Bioreceptors and analyte, like if it is an antigen antibody, the analyte will be antigen, and the receptor will be antibody. The bioreceptor will be antibody, and a transducers. that convert biological signal into a measurable electrical response. So bioreceptors, as I said, it can be organic or biological materials such as enzyme, antigen, microbe, hormone, or nucleic acid. For example, how this biosensor work basically. See, for example, you are fermenting, you are producing yogurt, okay, and you know your yogurt is produced from the milk, and how you will proceed? You will first boil the milk and then you will inoculate it, and then after incubations, it will turn to a set yogurt. This is very common method and everybody knows. So, what is the role of the bioreceptor? Or sorry, these biosensors. These biosensors let you are producing yogurt in bulk. I think around ton, or you can say a huge quantity, and then accidentally your culture was having salmonella. So we will keep a biosensor in that vessel or in that particular setup. Let salmonella has entered. So we will target some enzymes that is only secreted by the salmonella, or we will target some proteins which are only available if the salmonella is growing. Okay, so on the basis of those proteins and enzymes and some kind of metabolites, we will develop a counterpart. Like if it is enzymes, then we will take a substrate, and if enzyme is there, then enzyme will join with the substrate and we will give a color kind of thing. So that is a very deep story. Don't go in the deep. Just I'm trying to explain what biosensor is. In the biosensor, there is a bioreceptors. As I said, if it is an antigen, then our antibody will be the bioreceptors. In antibody, we will couple a antibody with a photoilluminators, or a, we can say, coloring compound. Okay, this let there are two part in that antibody. Part A is the antibody and part B is the coloring compound. So it will remain inactive until unless it doesn't couple with the antigens. As the antigen will enter, this antibody will couple with the antigen, and as they will couple together, it will give a light signal. That is a job of biosensors. Fine. Transducers can assume many forms such as electrochemical, optical, aquatic, depending on the parameters being measured. So further, this bioreceptors is convert uh, this after this there is a job of transducer. This transducer converts the biological signal into the measurable electrical response, and it will work on electrochemical, optical, or acoustic. So nowadays impedance sensor have been used. Impedance is something um, you might have studied about conductance. Okay, so every food conductance, and the opposite of conductance is impedance, or simply you can say it is a resistance. So when the food is contaminated with the microorganisms, so they affect the impedance of the food product. For example, initially the conductivity of the food product will be very less. As the microorganism will grow, they will break down the larger component into smaller component, and in between intermediate product will also form. As a result, the mobility of the charged particle will increase, or the number of the charged particle will increase in the system. Fine. So as a result, the conductivity will increase. As the microorganism will grow in the food, conductivity will the increase and impedance will decrease. So impedance based biosensors have been used since long. The recent one are the PDAs. The polydicetylylens are a class of linear conjugated polymer with alternating double and triple bond that can form vesicle or thin film which changes color from deep blue to red in response to different stimuli such as temperature, pH, and presence of biological molecule. Known nowadays, nanosensor and the nanomaterial based assays for detection of food relevant analyte including gases, small organic acid, and foodborne pathogens are in applications, or you can say the scientists are doing research. I'll take a break and then I'll come back. Please wait. So this is the schematic diagrams which show how this biosensor works. So the analyte species, this is the analyte, and here it will be biointerfacings. So here we can keep, let it is antigen, here we will keep antibody. So this antibody has traced, or you can say this antigens, then it will be converted into a, it will be converted into electrical signal with the help of transducer, and finally there will be amplification of the data, and then we can monitor it with the help of computer. So the second job of this intelligent packaging is second type of the intelligent packaging system is the providing more convenience. Fine. So for this there are two systems. One is the thermochromic inks. Thermochromic inks means these inks will behave differently at different temperatures. So we will make a barcode on the sample. If the temperature of that, let we have make a barcode on the food package, and if the food package has not been stored on a appropriate storage temperature, then there can be a widening of the barcode and we will not able to read it. So this will show the failure of the barcode. Okay, so this is the one example. We can read it. Can be printed onto the label or container which are to be heated or cooled prior to consumptions to indicate the ideal drinking temperature for the product. Depending on their compositions, the ink will change color at a specific temperature, and if the appropriate colors are chosen, hidden messages such as drink now or too hot become visible. The temperature abused product can be visually identified by a extra wide colored bar in the lower barcode with a which makes it unreadable at the checkout. Okay, so this is the story when it is doing something good. That we are having a kind of beer, and it is recommended to drink the beer. It should be at 10-degree temperature, so we can make a photochromatic ink photochromic label on that beer, and if as it reaches to the temperature 10-degree, then it will appear drink now. If it is a what you say now, this if it is a kind of anything that we drink by doing it hot, what is this? I think I have not seen any product that is packed and we consume it hot, too hot. Like same thing, we can say like that. There is a kind of any fruit juice, and if it has not been stored at a high appropriate temperature, or you can say that temperature of storage was very high. So at the high temperature, it will not give the appropriate sensory quality. So let the temperature of storage was recommended around 20 degrees centigrade, and accidentally it remained outside, and the outside temperature was 48 or 49 as it is nowadays in India. So if it reaches to 40 degrees centigrade, then it will appear too hot. Fine, I hope you understand. And this is something to check the this temperature abusive process. Like if the temperature has crossed the prescribed limit, then there will be a whitening of this barcode. Microwave doneness indicators, these are nothing but they are very, you can say, kind of facilitated to us. It is very difficult for us to assess what is the right time when we keep a product inside the microwave. Then sometime it is underheated, sometime it is overheated, and even the burning problem may takes place. Okay, now if you are packaging a product and if you say that please heat it before consumption, so and place it in the microwave for 2 minutes, 3 minutes, or at this particular watt, then it can help us that this is the right time to take out the product. So basically it is a label on the product, and when you keep it inside the when you keep the product inside the microwave, on the product package there is a label, and as the microwave heating prescribed time temperature heating it has achieved, then it will show that your microwave heating is means now your product is ready to consume. Microwave doneness means it is over now. So for that there is a prerequisite that we can use this label only for the product which are having showing the uniform type of the heatings. If there is a non-uniform type of the heating, then the hotter regions will trigger the doneness indicator long before the cooler region and has achieved an acceptable temperature. So the preferred location for a indicator to is the lid or the dome of the container directly above the food. Providing protection against the theft, counterfeiting, and tampering. this is very uncommon in the food, but it is generally used for the cloth and the electronic item. But nowadays people are using especially for the tempering, like what the people are doing. I can share the example of India. Let you are fond of drinking very good quality whiskey like Jack Daniel or Red Label whiskey. So what they do basically, they remove the lid, okay, and they empty the bottle, half them, they take half of the bottle, okay, then they fill it with a cheap liquor. So this type of you can say cheating is very common in India. So for that purpose we can use some intelligent packagings. Theft is nobody will, I think theft of the product is not that common. It will be something if we are producing something very valuable, like a Codiosep is the product, and if we have make a can, and on that can we can label with this you can say theft proof kind of sensors, fine. So because Codiosep is very costly, and during the transportation or during the transit it can be theft by somebody can steal it from you. So to reduce the incidence of theft and counterfeiting, holograms, special ink, dyes, laser label, electronic tags have been introduced. But their use in food packaging is minimal, again largely for the cost reasons. Intelligent tamper evident technologies are being developed based on the labels or seals which are transparent until the package is opened or tampered with, at which time they change color permanently. A word such as stop or open become visible. Like you have make a polythene, or you have packed the Codiosep in a pouch, fine, and we have provided a intelligent tamper evident seal on that pouch. In case of Codioceps, I was informed by some Bhutanese that there are two three qualities, A plus A B, like that. Means there are some poor quality, there are some very good quality. If somebody used to mix the poor and bad together, now just for the purpose of earning some money, undue benefits, so in that case, if that tag is there, then it will appear like that it was opened. This seal will, if somebody has opened it, then it will appear open. Labels or seals which release a dye on being ruptured are also in development, but their widespread use on food packages unlikely for the cost reasons. So very obvious that generally the food we consume are not that costly. That's why we cannot afford this type of the labels. But for the Codioceps, and as I said, the good quality of wine or whiskies, and something that is, you can say, life-saving thing, we can use such type of labels. Safety and the regulatory issue. So if we talk about the intelligent and active packagings, there can be some safety and regulatory issue, but there is no specific method existed in the national and international legislation to determine their suitability in the direct contact with the food. European Union regulations announced that the legislation which applies to the traditional packaging material has been applied to active and intelligent packaging. Same thing. The use of some type of active packaging raises the safety issue. Somebody says, na, active packaging is good for the food product. What can be the safety issue? Like you have put a oxygen sequencer inside the food product. So this oxygen sequencer will absorb the whole oxygens, and it will create an anaerobic condition, and this will be a preferable environment for the growth of Clostridium botulinum. So this is the safety regulations. Second is the inclusion of antimicrobial agent. Like you are storing the meat, and in the meat there are some some microorganisms were there on the skins, and we have stored in the film. It was having antimicrobial agent, and that antimicrobial agent film will be in contact with the packaging material, may result in the change in microbiological ecology of the food, and the type of the microorganism present on the food will be different from the same food packaged in a conventional manner. I mean to say that, let you are packaging meat. One meat has been packed in a very simple wrapping in which there was no antimicrobial agent, and you have kept in the frozen storage, and it was safe to consume. Other than this, to be overcautious, you are using another membrane that is having antimicrobial agent. So here the question or concern is that this antimicrobial agent film can change the ecology of the microorganisms, or the microorganisms that are present on the food which is stored conventionally will be different from the food which has been stored with the antimicrobial agent film. Antimicrobial film which only inhibit the spoilage microorganism without affecting the growth of pathogenic bacteria are also raise food safety concerns. Yeah, means there are antimicrobial films can inhibit the spoilage microorganisms, but they fail to answer some pathogenic bacteria. What about these pathogenic bacteria? How we can control them? European Union regulation defined the active materials and the article as the material and the article that are intended to extend the shelf life of the maintain and improve the condition of the packaged food. So these are some safety regulatory issues. If we are using these intelligent and active packagings, then there can be certain problems. So it is bit complicated part. I have selected only the simple versions. So it's all about, I think, from my side. Thank you very much.